In this video, we will be going over what is known regarding the pathophysiology of inflammatory bowel disease, or IBD. And this is not fully understood and is very complex because IBD includes two very distinct diseases. But they both have a common theme of autoimmune-associated GI inflammation and tissue damage. So the first disease that falls under this umbrella of IBD is Crohn's disease. And this can involve any segment of the GI tract. And the inflammation and damage is often discontinuous with this sort of skipping pattern. And these lesions can be transmural, meaning that they go through all of the GI layers, so all four of those layers that we've talked about in previous videos. Now, the second disease that falls under this umbrella of IBD is ulcerative colitis. And a lot of people will abbreviate this as UC, and I'll probably refer to it as UC throughout the video. And this is confined to the colon, and in some cases, only the rectum. And this inflammation is continuous, primarily only affects the mucosal and submucosal layers of the GI tract. So it is believed that some sort of environmental or genetic factor, and this causes a loss of tight junction integrity and leakiness. This allows bacterial antigens from the gut flora, which may involve some level of dysbiosis, to interact with antigen-presenting cells, or APCs, like dendritic cells or macrophages, which will phagocytize the bacterial antigen originating from the gut and release a number of cytokines, like tumor necrosis factor alpha, or TNF alpha, interleukin 12, and interleukin 23, and others to promote inflammation and activate and recruit other immune cells. So the antigen-presenting cell will also present that bacterial antigen originating from the gut flora with Th cells or T helper cells to trigger their activation and proliferation to become Th0 cells. These Th0 cells then differentiate based upon their cytokine environment. For instance, in Crohn's disease, Th1 cells are an important component, and Th0 cells differentiate via cytokines like interferon gamma and interleukin 12. Differentiated Th1 cells will then go on to promote cell-mediated response, like through the secretion of tumor necrosis factor alpha again and interferon gamma. And these will recruit and activate macrophages to promote inflammation and secrete the same cytokines to perpetuate the signal and continue the inflammatory process that leads to damage. Th17 cells, which are also an important component in Crohn's disease, are activated by interleukin 12 and 23 to also promote inflammation and damage. However, in ulcerative colitis, Th2 cells play a much more pivotal role. And Th0 cells differentiate into Th2 cells in an IL-4 or interleukin-4 environment. And then these cells will then secrete other cytokines to recruit lymphocytes like B cells, leading to a more humoral immune response. And one thing to note is that this recruitment of immune cells to the site of GI inflammation, whether it be these original T helper cells or following the secretion of cytokines from these Th1, Th2, and Th17 cells, the recruitment of these immune cells to the site of inflammation to begin this cascade requires the interaction of integrins, which I'm showing you on these immune cells here, like alpha-4 beta-7 integrin. And these integrins are very important for T cell homing to the gut. And once these integrins interact with their integrin receptor, they can then move from the peripheral circulation and be recruited to the site of inflammation in the GI tract to begin this cascade. You'll notice that there is another population of T cells shown in the schematic that we haven't talked about yet, and these are the Treg cells or the T regulator cells. And their role is to limit the immune and inflammatory response in the GI tract and keep this in check. Because the symbiotic relationship between the human body and the gut microflora requires some degree of sampling of these bacterial antigens to ensure the immune system can tolerate the normal GI flora and not sense it as an infection. So again, there has to be the special balance between effector and regulatory T cells to maintain this relationship. But as you can tell, there is an imbalance in the T cell response in the GI tract in individuals with IBD. And this may also be coupled, like I said before, with an unbalanced gut flora or a dysbiosis. And together this leads to that pro-inflammatory and autoimmune response. So the takeaway from this slide is that IBD encompasses two distinct diseases that appear to have different mechanisms and presentations, but they both have a disturbed balance between anti and pro-inflammatory signals, with this consequent migration of leukocytes to the GI tract, and this is coupled with an exaggerated or unbalanced T cell immune response. Moving to the next slide. Now this overview of IBD is somewhat simplified, so know there are a number of other cells and cytokines involved in Crohn's disease and ulcerative colitis, but I included several key cytokines by name to help guide us through the therapeutics indicated for the treatment of IBD. Now, just as a disclaimer, I do want to point out that some agents are indicated for Crohn's, some for ulcerative colitis, and some for both. Or some may be indicated for mild to moderate disease, others moderate to severe disease. And I will not really focus on indications as we go through these, only mechanisms of action and important pharmacological considerations, because we have a lot of agents to cover in this short video. So we will start off with the agents with the most straightforward mechanism of action, being the antibodies or the mAbs. And as a hint, all of their names will end in mAb, and this just means they are monoclonal antibodies or antibody fragments. So the first agents we'll talk about will target tumor necrosis factor alpha or TNF alpha, and this is a key inflammatory cytokine that you have seen in other videos, and it is a potent inducer of inflammation and damage in the GI tract and is released by a number of immune cells involved in IBD. So the antibodies that target tumor necrosis factor alpha are infliximab, which is Remicade, adalimumab, which is Humira, golimumab, which is Simponi, and the last one, which is the least preferred of these agents, is certolizumab, Pegol, and this is also known as Cimzia, and this is one that is a pegylated antibody fragment. So the mAbs will all bind and either neutralize soluble tumor necrosis factor alpha and or bind the transmembrane form, which can be found on some immune cells. And when it binds the tumor necrosis factor alpha that's found on immune cells, this can induce apoptosis. This will lead to reduced inflammation and circulating immune cells to improve IBD symptoms. The next agent is ustekinumab, and this is a monoclonal antibody that's also known as Stelara, and it is against interleukin 12 and 23. And it will lead to a similar cytokine neutralization and a dampened inflammatory response in the GI tract. So the last mAb that we're going to discuss is vedolizumab, or Entyvio, and this is an antibody to the alpha-4 beta-7 integrin. I mentioned that was essential for the homing of T cells to the GI tract. So by binding these integrin molecules on the T cells, it blocks their movement into the GI layer, so they are unable to participate in that exaggerated T cell response in IBD, thereby reducing inflammation as well. Moving to the next slide. Now, there are a number of concerns that are common across all of these antibodies. So first, they are all proteins. So they cannot be given orally, otherwise they will just be degraded by the harsh environment in the stomach, and they're so large because they're monoclonal antibodies that they wouldn't be absorbed anyways, even if they made it. So they are all either administered via IV infusion or sub-Q injection. Also, because they are proteins and huge molecules, there are a lot of opportunities for the body to recognize different epitopes on the antibody as foreign, and this leads to hypersensitivity reactions. Like anaphylaxis, and some need pre-medications before they can be administered. And this risk of hypersensitivity reaction and anaphylaxis varies based on what kind of antibody we are talking about. So this actually brings me to antibody naming. So let's talk about this for a second. So let's use golimumab as an example. So the last three letters of each antibody are mab, and this is just to indicate that they are a monoclonal antibody. So the first few letters of each antibody name are unique to that particular antibody. And then if the middle antibody name contains an L or an LI, then the antibody is against an immunomodulatory ligand. But if we look at ustekinumab, for instance, for Stelara, it has a Ki in the middle, and this indicates that it will bind an interleukin molecule. But now, if what follows is a U or an Mu, in the case of golimumab, then this antibody is human. And human antibodies like golimumab and adalimumab, being Humira, have the lowest potential for these hypersensitivity reactions and anaphylaxis because they are a completely human antibody, so less likely to be recognized as foreign. If these center letters are a ZU, like in certolizumab or vedolizumab, this means that the antibody is humanized and contains a very small portion in the FAB region of the antibody. Remember, we have this. We have the FC region of an antibody and an FAB region. So in the humanized antibodies, there's a very small portion that is not human. For instance, with Cimzia or certolizumab, this is 95% human and 5% murine. And this will have a moderate risk for a hypersensitivity reaction because, again, there's going to be a low chance for anything being recognized as foreign, but there's definitely that murine component. Now, if the central region is an XI, like in infliximab or Remicade, this means that that antibody is a chimeric antibody. And this will have a human FC region, but the FAB region will be from another species, like with infliximab, which has a murine FAB region. And this has the most foreign protein component, so this is going to be the highest risk for infusion reactions and hypersensitivity reactions. Now, moving on to other commonalities in terms of ADRs amongst these antibodies. So because all of these agents modulate the immune system, many will have a risk of serious infection, risk of activating a latent infection like tuberculosis, and malignancies like lymphoma. And this is because you are altering the normal checks and balances of the immune system. I do want to point out that Entyvio or vedolizumab would theoretically have less risk of this generalized immunosuppression because, remember, has this gut-specific mechanism. But there's still going to be that slight risk if there's any off-target effect. So now this covers the good majority of the agents used for the treatment of IBD, but now we are going to transition and talk about other immunomodulators that are not antibodies, and their mechanisms are a little bit more complex, and they exert their effects internally on immune cells. So we will zoom in on a T cell, but know that these agents can have effects on a broad range of immune cells. Moving to the next slide. So for the first group of agents, we're going to discuss the aminosalicylates, which you may also see as the 5 aminosalicylates or the 5 ASAs. And these are normally only used for mild to moderate ulcerative colitis. And these agents include sulfasalazine, mesalamine, and others. Now, the structure of some of these agents ensures that they are delivered in their prodrug form to the colon, where they are cleaved by colonic bacteria to give their active metabolite, to exert their effects locally to reduce inflammation. Or these agents are formulated to be delayed release or pH protected, so that they can reach their site of action in the colon. Now, these agents look and sound a lot like aspirin, which is also abbreviated ASA, and these are all salicylates. And if you remember, the mechanism of action of aspirin is it's a cyclooxygenase inhibitor. So remember, membrane phospholipids are converted to arachidonic acid by phospholipase A2. Arachidonic acid is then converted to a series of different prostaglandins, a number of which are pro-inflammatory. Some are protective, but some are pro-inflammatory. And arachidonic acid is converted to prostaglandins by cyclooxygenase enzymes like COX-1 and COX-2, which, remember, COX-2 is the inducible form at sites of inflammation. So aspirin inhibits these COX-1 and COX-2 enzymes. So for a long time, it was thought that these 5 ASAs, or the aminosalicylates, similarly inhibit cyclooxygenase enzymes, inhibiting the production of prostaglandins. But remember, some of those protective prostaglandins will be inhibited too. But these aminosalicylates that are used for IBD don't really seem to make IBD worse, which is what you would expect with aspirin and other nonsteroidal anti-inflammatory drugs that inhibit these COX-1 and COX-2 enzymes, inhibiting the good prostaglandin synthesis, like we talked about with GI ulcers and the peptic ulcer disease video. So now it is thought that these agents inhibit the conversion of arachidonic acid to leukotrienes, which is another group of pro-inflammatory eicosanoids like the prostaglandins. And these aminosalicylates achieve this through the inhibition of lipoxygenase, which is responsible for this conversion of arachidonic acid to leukotrienes. So a number of other mechanisms have been suggested as well. So the exact mechanism of action of these aminosalicylates is somewhat unclear, but we do know that these agents produce general anti-inflammatory effects locally in the colon. And these agents are generally well tolerated. So moving to the next slide. So moving on to the corticosteroids, specifically the glucocorticoids. And this is the video where we will finally dive into the details regarding steroid therapy. So these agents include things like prednisone, methylprednisolone, dexamethasone, hydrocortisone, etc. And these are potent anti-inflammatory agents. But they come with a lot of concerns, so these are usually reserved for acute exacerbations of IBD. So when these glucocorticoids are administered, agents will come in and bind the glucocorticoid receptor. And the glucocorticoid receptor, once it's bound, will then translocate to the nucleus to glucocorticoid response elements and target genes. This will either inhibit the transcription of pro-inflammatory genes or promote the expression of anti-inflammatory genes. And one example would be lipocortin, which can then come in and inhibit phospholipase A2, thereby preventing the formation of arachidonic acid and downstream pro-inflammatory mediators like the leukotrienes and the prostaglandins. The glucocorticoid-bound receptor can also recruit HDAC2 or histone deacetylase 2 to modulate gene expression through deacetylation of histones, causing pro-inflammatory gene repression. This will lead to reduced lymphocyte numbers, activation, proliferation, migration, etc., to help dampen that immune response in IBD. While they are potent, they come with a lot of adverse drug reactions and concerns. First, they are immunosuppressants, so the risk of infection is there, but because they are steroids and look a lot like cortisol, they are going to throw off a number of steroid hormone feedback loops. So some of the major adverse drug reactions to be aware of are infections, like I said, retention of water, and salt. So there will be a significant risk for edema and hypertension. There is a risk of peptic ulcer disease, adrenal suppression, again, feedback loops, hyperglycemia, psychosis, or general mood changes, Cushing's, cataracts and glaucoma, osteoporosis, and poor wound healing. So for all of these reasons collectively, generally the dose of the corticosteroid should be limited and the duration should be limited. But these agents have to be tapered to avoid corticosteroid withdrawal symptoms, because remember, again, we have the adrenal suppression. We need to taper them off of this agent to ensure that their adrenal has time to restart making cortisol to compensate. So moving to our next slide, being the antimetabolites. So our first group of agents are going to be the thiopurines, which are either 6-mercaptopurine, which is sometimes abbreviated as 6-MP, and azathioprine, which is sometimes abbreviated AZA. And azathioprine is a prodrug which is converted to 6-MP in the body. Now these agents look a lot like purine bases, so they are fed into the purine biosynthesis pathway, which we covered in the gout video, to inhibit a number of enzymes involved in that pathway. And these agents will competitively inhibit those enzymes from synthesizing new purines, and this depletes the purine nucleotide pools. And this will inhibit DNA synthesis and immune cell proliferation, contributing to that anti-inflammatory effect. So methotrexate, which is sometimes abbreviated MTX, is also an antimetabolite, but instead mimics the structure of folic acid. And methotrexate competitively inhibits tetrahydrofolate reductase, which is the enzyme that is responsible for converting folic acid to its active form. And then the active form of folic acid can participate in purine and thiamine synthesis. So methotrexate will inhibit this process, which will similarly lead to depleted nucleotide pools and inhibition of DNA synthesis and inhibition of immune cell proliferation. Now these antimetabolites are general immunosuppressants, so there will be a risk of infection and malignancy, but also because they interfere with cell proliferation, they're going to lead to ADRs that are associated with rapidly dividing cells. And these cell populations are going to be in the skin, the GI tract, and the bone marrow. And the use of these agents could potentially lead to things like alopecia and rash, nausea, vomiting, and diarrhea if they don't already have diarrhea, and myelosuppression. Because of these risks, these agents are normally seen with severe or resistant IBD. So moving on to the next slide. So the last agent we're going to talk in detail for today, which you have probably seen in commercials, is Xeljanz or tofacitinib. And this nib ending signifies that it is a small molecule inhibitor. So tofacitinib inhibits JAK kinases or Janus kinases, and these are intracellular enzymes that are coupled to cytokine receptors. So when tofacitinib isn't around, cytokine binding will lead to the phosphorylation of STAT or signal transducers and activators of transcription molecules. And this phosphorylation of STAT by JAK triggers their dimerization and nuclear translocation to lead to the transcription of pro-inflammatory genes and other positive regulators of immune cell function. But with tofacitinib, JAK is inhibited, preventing this phosphorylation of STAT, so these genes are never expressed, preventing proper immune cell function and the production of these inflammatory mediators. The adverse drug reactions and concerns are very similar to the other agents, where immunosuppression leads to an increased risk of infection and malignancy. And there is also going to be effects on some of those rapidly dividing cellular populations that relies on JAK-STAT signaling. There is also a risk of major cardiovascular events with Xeljanz, and it is pretty expensive. So this is usually only used after failing another agent. So moving on to the next slide. In summary, the pathophysiology of inflammatory bowel disease is complex, as it encompasses two distinct diseases, being ulcerative colitis and Crohn's disease. Equally as complex is the treatment of IBD, which includes a number of drugs that have been around for several decades, like the corticosteroids, the aminosalicylates, and the antimetabolites, as well as a number of newer revolutionary therapies, like all the MABs, the tyrosine kinase inhibitors. However, once you begin to place these agents into bins based on their targets or their adverse drug reactions, this long list of agents and information becomes much more manageable.